teaching in a very well-established institute, that is Gaining Apex. Today I want to discuss with you a very important biology chapter, that is flower. Flower is a very important concept in bio. Today I am covering the following concept during my lecture. First is parts of a flower. Second, microsporogenesis. Third, structure of male gametophyte. Fourth, megagametogenesis. Fifth, structure of female gametophyte. And last, very important topic, that is fertilization in angiosperm. These 6 topics are very much important related to flower. Now, I am starting from first, parts of a flower. As you know that flower is a reproductive part of a plant. Flower has basically 5 parts. First is apicalyx. Second one is calyx. Then corolla. Then androecium. After that, last part is gynoecium. The apicalyx only present in Solanaceae family. It is only a protective in function. Calyx, other name is sepal or green part of a flower. It is also protective in function. Corolla, colorful part or called petals, which interact the insect as well as human. Androecium, other name is stamen and commonly called male reproductive part. The last one is gynoecium, other name is pistil as well as carpel, and this is for female reproductive part. First 3 are accessory parts of a flower, but these 2 are essential part of a flower. They help in reproduction of a flower. I will again repeat, there are 5 parts of a flower. First is apicalyx, calyx, corolla, androecium and then gynoecium. These 3 first parts are only for accessory. They never play any role in reproduction, and last 2, that is androecium or gynoecium, take part in reproduction. Now I am taking a flower that is hibiscus, China rose or bisexual flower. These one, a very small, is called apicalyx. This is a main feature of Solanaceae flower. Only Solanaceae flower have apicalyx layer. Then this one is called sepal, green layer of a flower, which I am separating out now. Then there are colorful petals. These are called corolla. And after that, these yellow colors, they are called stamen or anther. This is a filament, these are the anthers, male reproductive part. Now I am eliminating the male reproductive part, and then we get a female reproductive part, stigma, style, ovary. This is a female reproductive part, and here I will also try to explain you the ovule of a flower. This is a ovule of a flower. Before microsporogenesis, I want to tell you about male reproductive part, that is stamen. You know the structure of stem and then only you will know about
Speak
microsporogenesis.
loudly.
This is an anther and this is a filament, male reproductive part. Anther is a fertile part which take parts in reproduction. And this is a fertile part which doesn't take reproduction. And now, microsporogenesis. It is a euspongiate type. Euspongiate type means that starting from archisporial cells, anything in bio which starts from archisporial cells are known as euspongiate type. These cells firstly divide periclinally and form two layers. First one is outer and the second one is inner layer. Outer layer is called parietal cell and the inner layer is called sporogenous cell. Parietal cell further divide periclinally and form three layers. First one is endothecium, second one is middle layer and the third one is tapetum. These three layers help and provide the nutrition to the sporogenous cell. Sporogenous cell divides and form MMC, that is spore mother cell. And here it is divide meiosis and form 4 microspores. I will again repeat, microsporogenesis is a euspongiate type. So euspongiate type means anything in bio which starts from archisporial cells or initials that are called euspongiate type. Archisporial cells first divide periclinally to outer and inner layer. Outer layer then form parietal cells and these divide periclinally and form three layers: endothecium, middle and tapetum. And this parietal cell is diploid, that is 2n. Further, inner layer forms sporogenous cell, that is also a 2n, which will form MMC spore mother cell, which is also a 2n. Then after meiosis, 4 microspores are formed. They are called N, means haploid. Now these 4 microspore cells I will draw. They form a tetrad. Tetrad means 4 cells in a single structure. That is known as tetrad. Now I will erase. Now in tetrad there are 4 cells, 3 cells degenerate and only 1 cell form male gametophyte. One cell divides mitotically to form one vegetative cell and the other one is generative cell. One vegetative cell form pollen tube and generative cell form gametes. That is a structure. That is a generative cell and that is a vegetative cell. And generative cell form pollen tube and vegetative cell form only gametes. These are formed by generative cells and these are formed by vegetative cells. This is a structure of male gametophyte. You will remember this structure for reproduction. I will again repeat. 3 cells degenerate, 1 cell form male gametophyte. It will divide mitotically to form 1 vegetative cell and 1 generative cell. 1 vegetative cells form pollen tube and the generative cell form gametes. Here is a structure of male gametophyte. This is a generative cell, this is a vegetative cell. Vegetative cells form the pollen tube as I shown in with red marker, and generative cell form the gametes, and this is a structure of male gametophyte. Now I will start with megagametogenesis. Before megagametogenesis, you will know about the structure of female reproductive part, that is pistil. Here it is 3 parts: stigma, style, and last one is ovary. We will discuss the formation of this part only. Here it is also starts euspongiate type. Now you will familiar the term euspongiate, then anything in bio which formed from archisporal initial is known as eusporangiate type. It will also divide periclinally into 2 parts, that is outer and inner. Outer one is called peritial cells and inner one is called sporogenous cell. Again, sporogenous cell by meiosis form 4 spores that are N, that is haploid. From these 4, first 3 will degenerate and last one act as a female gametophyte. I will again explain you female reproductive part, stigma, style, and last is ovary. We will discuss about ovary because here it is the reproduction takes place. Again, formation of ovary is euspongiate type, which means it starts from archisporal initials. These cells divide periclinally to form outer as well as inner layer. Outer layer again form peritial cells and inner layer forms sporogenous tissue. Then from meiosis it forms 4 spores. These 4 spores, 3 will degenerate and only 1 will form female gametophyte. Now we will discuss about the female gametophyte. Last we do that, there is one female gametophyte cell, that is this. It will divide mitotically first to form 2 nucleus inside a sporangium. Then it will again divide mitotically and form 4 cells in a sporangium. It will again divide mitotically and form 8 cells in a sporangium. Now, from these 8 cells, 3 move opposite direction and form different structure in it, and 2 will come in the center. These are called antipodal cells. These 2 are called polar nuclei, and these are called synergids, which only provides the nutrition to the eggs, and this center one is called egg. This is a structure of female gametophyte. I will again repeat. This is a main cell. from where megasporogenesis takes place, it will go under 3 mitotic division to produce 8 cells in a sporangium. First it will form 2, later on 4 and later on 8. From these 8, 3 cells moves in an opposite direction and form different structure, and 2 will come in the center. The 3 upper side is known as antipodal cells, and the 3 larger one in which 2 are synergid and center one is called egg, and center 2 cells are called polar nuclei. But remember, this all occurs in a sporangium. ये female gametophyte. This is a whole structure of female gametophyte. For reproduction, we should remember this structure also. Last very important topic, that is fertilization. Fertilization is a very important process for production of new flowers. First of all, I will draw female gametophyte. We will make this diagram that how this gametophyte is formed. These 3 are called antipodal cells. These 2 are called polar nuclei. And last, these 2 are called synergids. And the front and center one is called egg. Now you also remember male gametophyte, which we also know about how microsporogenesis takes place. This is a male gametophyte and this is a female gametophyte. Now this part have to reach this part through the agent, and the process is called pollination. It may be insect, it may be through air, any contact. Now this male reproductive part could reach this part, absorb water, swell up and form a pollen tube, which is a main entry to female gametophyte. And when these male gametes through this pollen tube reaches to the female gametophyte or the main part, that is egg. These are 2. I will show the movement to you that these are slipped towards the pollen tube and reaches the egg. One male gamete fuses with egg to form zygote, and there is one male gamete which fuses with polar nuclei and form endosperm, and these both form double fertilization. Very important topic. What do you mean by double fertilization? I will again explain. This is a male gametophyte, this is a female gametophyte. This reaches to stigma point of female gametophyte. Here it absorbs water, swell up and form a pollen tube. Through tube, it reaches to the female reproductive part, that is ovary. Here it first fuses with the egg to form zygote. Then it fuses with 2 polar nuclei to form endosperm. Here the fertilization takes place in 2 times. First time it forms zygote and second time it will form endosperm. So 2 times fertilization is called double fertilization. Now I will again explain you by a flow chart so that you remember easily what do you mean by double fertilization. Male gamete plus egg will form zygote. Male gamete plus 2 polar nuclei endosperm. Here, endosperm and zygote, two products are formed. This is called double fertilization. So, I clear all the concept related to flower. So, today lecture is closed.